In this video, we'll be talking about the biosynthetic phase. Now, we have studied so far that the light reaction which takes place in presence of light has the results, the outcome of it is the few products which are being obtained. And the products which are obtained during the light reaction are ATP, which is produced in the stroma. So the ATP is produced in the stroma and it is produced with the help of an enzyme, that is ATP synthase. Another product obtained is NADPH. NADPH2. It is produced towards the stromal side. Stromal side of thylakoid cells. So thylakoid membrane towards the stromal side and later released into the stroma. The enzyme which is involved over here is NADP reductase. The third product which is obtained during the light reaction is oxygen, and it is a byproduct. This byproduct is produced in the lumen. It is produced in the lumen of thylakoid membrane. Now, these three are the products of the light reaction. Out of these three products, the oxygen, it diffuses out from the chloroplast. That is, it diffuses into the chloroplast. The NADPH and the ATP, which are produced in this reaction, are utilized to synthesize sugar. So they are utilized to synthesize sugars by the plants, and therefore it is known as biosynthetic phase. Since we are using the products of the light reaction to produce sugar, therefore it is known as biosynthetic phase. Now, this process, that is the formation of the sugar, does not directly depend upon the presence of light, but it depends upon the product of the light reaction. These two are the products of the light reaction. So we are not, this process, biosynthetic phase, is not dependent directly on the light for it to happen, but it depends upon the products which are obtained during the light reaction. And also it uses carbon dioxide and water. So since it does not require the process, biosynthetic process does not require light. So apart from ATP and NADPH2, CO2 and water, they are used. So therefore, since it does not require light, it is used, known as also the dark reaction. This reaction is also known as dark reaction. So biosynthetic phase, the dark reaction, both are same. It is also called as light independent reaction. Light independent reaction. Now, the entire process of this this dark reaction was actually explained by a scientist, which is Melvin Calvin. So this scientist actually traced the entire process of, or the entire cycle, using a radioactive carbon isotope. So he utilized radioactive carbon isotope to study and trace the pathway. He used green algae and utilized this carbon 14 and traced, which led to the knowing of complete biosynthetic pathway, and therefore this cycle is also called as Calvin cycle. So therefore this cycle is also known as Calvin cycle. So we have various names: biosynthetic phase, the dark reaction, the light independent reaction, or the Calvin cycle. Now the pathway is actually it was completely traced, and initially it was found that the first stable carbon compound during the cycle, the first stable carbon compound which was formed was three carbon atom compound called phosphoglyceric acid, or we also call it phosphoglycerate. It is also known as phosphoglycerate. Now it is written as PGA, and we write it is a three carbon atom compound. Now it was initially believed that this PGA actually is formed due to reaction when carbon dioxide from the atmosphere combines with a compound having two carbon atoms, or having two carbon. Therefore, it was presumed that this carbon dioxide and two carbon form the three carbon compound, that is phosphoglycerate. But later they found that it is not so, and it was, instead of a compound with two carbon atom, it was not so. Instead there was a compound which was a five carbon atom. So actually the carbon dioxide combines with a five carbon atom compound called RUBP. RUBP is ribulose biphosphate. So it is this compound which was actually a five carbon compound which combines with carbon dioxide, and this reaction actually is helped by an enzyme which is known as rubisco. This enzyme was rubisco, which actually helped in addition of carbon dioxide or combining of carbon dioxide with a five carbon atom called as RUBP, or ribulose biphosphate. Now RUBP is a ketose sugar. It is a ketose sugar, and when this carbon dioxide is accepted by RUBP, this process, the combining of carbon dioxide with this five carbon atom compound, is known as carboxylation, because here the addition of carbon dioxide is taking place. Now the rubisco enzyme is peculiar, that is, its full form is rubisco is ribulose carboxylase oxygenase. This enzyme rubisco is a very important enzyme, and the working and nature of this enzyme depends upon external factors such as the temperature, the concentration of carbon dioxide also. So that in certain conditions it will combine with carbon dioxide, or at other times it may combine with the oxygen. And also this is the most abundantly found enzyme. Now, when we study the Kelvin cycle, or let us study the Kelvin cycle in detail, we find that the Kelvin cycle is clearly divided into three major steps, and each of the step will be explaining here. Now, the steps which are involved in the Kelvin cycle are: first is carboxylation. So it is the first step. The second step is the reduction, and the third step is regeneration of RUBP, that is ribulose biphosphate. So we'll be starting with the explanation, and over here you have to understand that we take to understand this cycle, we are taking 3 carbon dioxide molecules, but in reality the plants take only 1 carbon atom per cycle, because this will when we will study the cycle, this will help us understand that why we are taking 3 carbon dioxide molecules. So let us begin with the first step, that is the 3 carbon atom. So the 3 carbon atoms, or the 3 molecules, we say 3 molecules of carbon dioxide combine with 3 molecule of RUBP. Now this RUBP and the carbon dioxide, in presence of enzyme Rubisco, combine to form a product which is 6 molecules of phosphoglycerate or phosphoglyceric acid. Now it is also written as PGA. While moving through the cycle, we'd like to keep a track of the carbon atoms also to make you understand better. Now over here we have used 3 carbon dioxide molecules. So that means there are 3 carbon over here. Now the 3 molecules of RUBP, and remember it is a 5 carbon compound. So over here 3 into 5, we have 15 carbon combining with 3 carbon, that is the carbon dioxide, and here 6 molecules of phosphoglycerate are formed. As a result, each molecule is having, the PGA is a 3 carbon atom compound. So therefore 6 into 3, that makes it 18 carbon over here. Now this the Rubisco enzyme over here act as carboxylase, because it is helping in combining carbon dioxide with RUBP. Now we know that the first stable compound is phosphoglycerate or phosphoglyceric acid, and it is written as 3-PGA. Now it is written as 3-PGA over here because the carbon atom which is attached, the third is the number of carbon on which the phosphate group is attached. And now 6 molecules are synthesized by the combination, and each molecule having 3 carbon, because this is the first stable compound. So therefore 18 carbon are present over there. Now the PGA, that is this PGA, undergoes phosphorylation because it has phosphate at 3 carbon, and it undergoes phosphorylation. There are 6 PGAs, so therefore 6 ATP will be required for this, and 6 ADP will be produced, resulting into the formation of 1,3-diphosphoglycerate. So 1,3-diphosphoglycerate is formed. And this 1,3-diphosphate, there are 6 molecules of this are produced. So So here we have 6 molecules. So the number of the carbon remains the same. That is, still we are having the 18 carbon atoms. Now what happens is, the compound which is 1,3-diphosphoglycerate, it is same as phosphoglycerate, but it differs in having 2 phosphate, which is having one phosphate group is attached at first carbon and the second is attached at the third carbon atom. It has same number of carbon compounds. Now this undergoes reduction and we obtain PgAl. PgAl is actually phosphoglyceraldehyde. Over here, reduction is done, and this reduction is done with the help of NADPH2. So NADPH2 undergoes reduction and NADPH are formed. But here 6 NADPH2 are there, which are being reduced to 6 NADP, and then 6 inorganic phosphate are released. So this PgAl, I am writing the full form over here, that is phosphoglyceraldehyde. What is happening over here? We are getting 6 molecules of PgAl and the number of carbon atom is still the same, 18 carbon. Now, the reduction of acid, that is the phosphoglyceric acid, has been reduced to aldehyde over here, and 6 inorganic phosphate will be produced during the process. It is a 3 carbon atom compound. This PgAl is also a 3 carbon atom compound. So 6 into 3 we have 18 carbon over here. Out of these 6 molecules of PgAl, 1 molecule of PgAl will move out of the cycle and remaining 5 will continue with the cycle. So we have the 1 molecule which has come out of this, and therefore the 3C, that is, we write with red over here. So this is 1, since it is a 3 carbon atom compound, 3 carbon have moved out over here, and the remaining 5 molecules of PgAl will continue with the cycle. Now 3 carbon have been removed, so we have 15 carbon over here with it. Now when we talk of this part, the 3 carbon atom have been separated and 5 molecules will continue. Now each PgAl has 1 phosphate, that is total 15 carbon over here and 5 phosphate. PgAl is having 1 carbon. So if we see over here, there are 15 carbon and each PgAl has 1 molecule of phosphate. So 5 phosphate group over here. That is monophosphate. Now we get this much. This undergoes phosphorylation, that is addition of phosphate. Now before we continue with the cycle, let us understand what happens to this 1 molecule of PgAl which has separated out. This molecule is utilized to synthesize glucose. Whereas the 5 molecules will continue in the cycle. Now these 5 molecules, each having 1 phosphate, that is, it is monophosphate state. Now this will undergo the process of phosphorylation because here the 3 ATP will be required. So what is happening over here? The PGAL molecule will undergo phosphorylation and will form ribulose 5 phosphate. And why we are mentioning ribulose 5 phosphate? There are 3 molecules which are produced. Now, this is a 15 carbon atom. This is a 15 carbon atom, and during the phosphorylation, 2 inorganic phosphates are released. Here, what is happening when it is undergoing the phosphorylation process? 3 ATP will be required. Let us understand this. Why we are saying ribulose 5-phosphate? Already mentioned, the PGAL is having 1 phosphate over here. Now, 2 Pi are removed. That means 2 phosphate groups are removed. So, what is happening is 3 molecules of ribulose 5-phosphate are produced, and each molecule will be having 1 phosphate. So, it's actually ribulose monophosphate. And why we are writing ribulose 5-phosphate? Because each molecule has 1 phosphate group which is attached at carbon 5. Therefore, the number 5 is mentioned over there. So, 3 molecules are produced and they undergo the process of phosphorylation with the help of ATP, and where it changes into 3 ADP, and we get the 3 molecules of RUBP. Now, this cycle is also referred to as C3 cycle. Why we are calling it as C3 cycle? Because the first stable compound which is formed after the combining is a 3 carbon atom compound. Let us divide now this cycle into stages. This process of combining of carbon dioxide with RUBP in presence of Rubisco, this is known as carboxylation. This process is carboxylation. Here the reduction is taking place. So this is known as reduction. And third step, that is the formation of RUBP. Because it is monophosphate, we require 3 molecules of phosphate to form ribulose biphosphate. So here this is the regeneration of RUBP. So this is the Kelvin cycle which help us to understand the formation. Now you have to understand also that during this process we have taken 3 carbon atoms, but as initially I told you, there is only 1 carbon dioxide molecule which is taken up by the plant. So for the synthesis of 1 glucose molecule, we require 6 Kelvin cycles. Because otherwise, if we would have taken 1 carbon dioxide, it would have been difficult to separate out PGL as in fractions. So we have, for our convenience, we have used 3 carbon dioxide. So for 1 synthesis of 1 glucose molecule, there is a requirement of 6 Kelvin cycles. Now let us count how many ATPs are required over here. Now see there, 6 ATP, and there are 3 more ATP which have been used. 6 ATP over there and 3 ATP over here. So we have 6 plus 3 ATP which is equal to 9 ATPs. Now these 9 ATPs, and since we are using 3 carbon dioxide to understand the cycle, so here we have and 3 carbon dioxide in one cycle, so 3 carbon dioxide in the next cycle. So we have 18 ATP taking place or being utilized in a cycle, that is synthesis of for 1 glucose molecule. because we have, try and understand this. For synthesis of one glucose molecule, actually by the plant, since it takes one carbon dioxide, there are 6 Kelvin cycles to be to have to take place. But over here we have taken 3 carbon dioxide molecules. Glucose is a 6 carbon molecule. So 2 times the cycle has to be repeated, and therefore 6 ATP and 3 ATP. That means 9 ATP are used in one cycle, and this cycle is to be repeated twice for us to understand. So therefore 18 ATP are required to synthesize one glucose molecule. The NADPH2 which is required during this process, that is 6 NADP are required one cycle. So we have 6 NADP in one cycle, and we are understanding in terms of 2 cycle for the synthesis of one glucose molecule. So therefore 6 into 2, that means we have 12 NADPH2 which are required during the Kelvin cycle. So this Kelvin cycle is very important, which makes us realize the synthesis or how the formation of glucose is taking place.